In this short animation, you will learn about polymerase chain reaction, or PCR. PCR is a relatively simple and inexpensive tool that you can use to focus in on a segment of DNA and copy it billions of times over. PCR is used every day to diagnose diseases, identify bacteria and viruses, match criminals to crime scenes, and in many other ways. Here I'm going to take you through a virtual lab bench so you can see how it works. The human genome is made up of 3 billion chemical base pairs. Scientists often need to isolate a very specific segment of DNA from within a vast amount of genetic material. Since this segment is just one tiny piece of the genome, they need to make copies to have enough to work with. The polymerase chain reaction, or PCR, can generate 100 billion identical copies of a specific DNA sequence in a matter of hours. As the inventor of PCR explained, the technique requires no more than a test tube, a few simple reagents, and a source of heat. To perform a PCR reaction, you need DNA that has been extracted from cells. Because the purpose of PCR is to make more DNA copies, you don't need very much DNA to start the reaction. For example, you could extract DNA from a small sample of blood, skin, saliva, or even hair follicles. Start by moving your extracted DNA into a special PCR tube. PCR works by heating and cooling the solution over and over, so PCR tubes are designed for even heat distribution. Here I'm using the micropipette to extract the exact amount required. Next, add primer 1 to the PCR tubes. Primers attach to sites on the DNA strands that are at either end of the segment that you want to copy. They are powerful tools for copying very specific DNA sequences, since there is almost no chance that they will target the wrong sites. Primers are short pieces of DNA that are made in the laboratory. Since they're custom built, primers can have any sequence of nucleotides you like. In a PCR experiment, 2 primers are designed to match to the segment of DNA you want to copy. Through complementary base pairing, 1 primer attaches to the top strand at one end of your segment of interest, and the other primer attaches to the bottom strand at the other end. In most cases, 2 primers that are 20 or so nucleotides long will target just one place in an entire genome. Primers are also necessary because DNA polymerase, which is a naturally occurring enzyme, can't attach at just any old place and start copying away. It can only add on to existing pieces of DNA. Now I'm going to use the micropipette again and insert the primer. Now add primer 2, which will attach to the second site. Add nucleotides to your PCR tube. Nucleotides are the A's, the C's, the G's, and the T's that make up the DNA code. These genetic building blocks will be used to create billions of DNA copies. Last, you'll add DNA polymerase to the PCR tube. DNA polymerase molecules act like tiny machines that read the DNA code and then attach matching nucleotides to create DNA copies. This particular DNA polymerase has been specially selected to withstand the high heat of the PCR reaction. Now that the PCR tube is filled with all of the reaction components, you'll place it into a DNA thermal cycler. This machine can precisely heat and cool your tube at specific times during the next hour. These changes in temperature are crucial for making the reaction work. Inside your PCR tube, cycle 1 has begun. The thermal cycler heats up to about 95 degrees Celsius. That's 203 degrees Fahrenheit, which is almost boiling. At this temperature, the DNA double helix starts to unwind, creating two single-stranded DNA molecules. Now the thermal cycler cools down to 50 degrees Celsius. At this temperature, single-stranded DNA molecules naturally attempt to pair up. However, there are many more primer sequences than DNA strands in the tube. The primers crowd their way in and lock onto their target before the strands can rejoin. The thermal cycler now changes temperature to 72 degrees Celsius, which activates the DNA polymerase. When DNA polymerase locates a primer attached to a single DNA strand, it begins to add complementary base nucleotides. It continues until it gets to the end of the strand and falls off. Cycle 1 is now complete. The same 3 steps happen in cycle 2. The temperature is raised again to separate the DNA strands. The temperature is lowered so the primers will attach. And the temperature is raised again slightly to stimulate the DNA polymerase to copy the strand. During cycle 3, your desired products begin to appear. 2 strands that begin with one primer at one end and end with primer 2. These are the DNA copies of just the segment of DNA you have targeted. Although there are only 2 desired fragments at this stage, as you continue with the cycles, these products will quickly become the majority. At the end of cycle 4, you have 8 fragments that contain only your target sequence. Now you have 22 fragments that contain only your target sequence and only 10 longer length copies. After 30 cycles, there are over 1 billion fragments that contain only your target sequence and only 60 copies of the longer length molecule. You now have a solution of nearly pure target sequence.